using a mixture design. So firstly, let's begin with some introduction. When people are saying RNA-seq, usually they are referring to the second generation short-read RNA-seq because the sequence, the materials are fragmented during the library preparation. So we are usually not able to get the full-length RNA sequence during this using this technology. So the data is also called short-read data. While for doing preprocessing for the short-read data, the first step is usually to map the reads to the reference genome or transcriptome so that we can know where does the reads come from. And after that, we can perform quantification on either like gene level, transcript level, or exon level so that we can know the abundance of each gene or transcript or exon. And after that, we can get a count matrix where we can know the expression of the genomic features in each sample. And by having this table, we can compare the expression level of the genomic features between different samples that are like under different treatment or other experimental groups. So we can know which genes or transcripts are turned on or off in different groups. Well, we are doing study like that for if we are doing gene level study, then all of our analysis are based on the genes. However, there's a molecular mechanism called alternative splicing that for genes that are containing more than one exon, some of the exons are not always expressed and used in the transcripts that actually they are actually transcribed that so that one gene can form multiple transcript isoforms. But when we are studying the on the gene level, all of our counts from the different isoforms are truncated into the same gene. So we will lose the higher resolution information on the transcripts. So that's why we need to study on the transcript level. Well, if we're using short-read RNA-seq for such kind of analysis, then because the reads are relatively short compared to the transcript, and usually it doesn't contain or contain only limited information for the exon connectivity. So when we are mapping the short reads to the transcript, there is a problem that sometimes the same read can be assigned to multiple transcripts. So there comes a transcriptome mapping uncertainty when we're using short reads to do the transcript level analysis. Well, now we have the third generation sequencing technology. So for example, nanopore sequencing is one of the technologies where you can get where it works by having the currency signal when the RNA or DNA molecule goes through an biological nanopores. So the currency information will be decoded to the sequence, and the sequence can be pretty long because it doesn't need to get fragmented during the library preparation. And the third generation sequencing is thus also called long-read sequencing. While comparing long-read versus short-read sequencing, the advantage of using long-read is that it's not fragmented, so we can possibly get full-length read that can cover the full-length transcripts. So it can, in theory, it can reduce the transcriptome assign ambiguity in the analysis. However, there are still some disadvantages of using long read. So for example, the sequencing accuracy of using long read is lower comparing to short read, which means in the data we get, there are a higher possibility of us to having some bases that are falsely detected to be other bases, or there may be some false indels. And also comparing to the short read technology, currently the long read technology, the number of reads we can get per flow cell is slower, which means if we are using the same number of flow cells to do the experiment, then the number of reads we can get is lower comparing to the short reads. And for long read transcriptomic data analysis, usually we also start the analysis workflow by mapping the reads to the genome or the transcriptome. And if we map the reads to the genome, there is an additional step called isoform detection. That's something that is not that easier to be done with short read. That's to do isoform detection, then we can detect if there are any novel transcripts that were not annotated but exists in the data. And after detecting the novel isoforms, we can map the reads to the transcripts that we detected in the isoform detection step. But we can also skip them, and if we have a reference transcriptome, we can also directly map the long reads to the reference transcriptome. And then with the reads mapped to the transcriptome, we can perform transcript level quantification. And with the count matrix on the transcript level, we can do differential analysis like differential transcript expression analysis, DTE analysis, or differential transcript usage analysis, DTU analysis, to study the differential splicing. Well, in this study, because there is a lack of suitable data to perform benchmarking for the tools, and also there are a lot of tools already available now. So in this study, our aim is to benchmark the long-read RNA-seq transcript level analysis tools, and we will focusing on the isoform detection, DTE and DTU tools. And you may wondering what's the difference between DTU analysis and DTE analysis. So here is an example where we assume there is a gene with two isoforms. Well, the two isoforms are expressed at different level, but if you look at the proportion of the isoform usage, you can find that they are also in the two different groups, they are used in the same proportion. So in this case, both of the two transcripts should be detected to be DTE transcripts, because for DTE analysis, we are focusing on the expression level of each transcripts. But for DTU analysis, we are focusing on the proportion of the transcript usage comparing to all the different transcripts from the same gene. So in this case, this example gene should not be DTU. So there are both methods to study the differential splicing, but they are focusing on different aspects. Okay, now let me explain. Since we want to compare the methods, then we need to have a suitable data set that contains some ground truth. Then let me introduce my dataset design. So firstly, we introduce ground truth to our dataset by using sequence spike-ins. So the spike-in contains 76 artificial genes, and some of the genes are also containing multiple different isoforms. And we know all the sequence of the isoforms, the genes, and also their abundance. And some of the transcripts or genes are designed to be differentialists, having different abundance. So since we know all the ground truth, we can use them to ask the known truth to detect whether, for example, whether our isoform detection tools are working well, or whether our DTE and DTU tools are working well. And other than the sequence spike-in, another way we use to introduce ground truth is using a mixture design. So the idea comes from a previous study on bulk short-read RNA-seq, that we choose the 2 different cell lines, and the cell line samples were mixed into different proportions as showed here. And a characteristic of the design is that although if, like, for example, for here we have different proportion, so we can have different comparisons of the samples. For example, we can compare the pure cell line samples, and also we can make comparisons between the in-between mixtures. And the characteristic of the mixture design is that the same set of genes must be DE between any pair of samples, but the fold change are different. For example, the genes that are differentially expressed in the comparison 1 should also be differentially expressed in the other comparisons. But the fold change in the comparison 2, 3, and 4 should be smaller than the comparison 1. So we can treat the DE we found in the comparison 1, where we are comparing the pure samples, as the ground truth, and we can compare the DE we detected in the other comparisons. So we can use this method to estimate the power and the false discovery rate of the methods. And because it will cost a lot to sequence all the in-between mixtures in real experiment in lab experiments. So here we used the data from the previous study as a pilot experiment, that we mixed the reads from the pure samples into the same proportion as the lab-derived mixture samples. And after that, we used the same methods to quantify them, and we did the preprocessing, and we made an MDS plot. And on that plot, the lab-derived samples are in the circles, while the in silico mixtures are in the triangles. And you can see that the in silico mixtures are performing quite similarly than the lab-derived samples, which suggests that our in silico mixture strategy works, that the in silico mix, we use this method to recover we can generate data that are quite similar to the lab mixture. So we used this strategy in our dataset design. So finally, we choose the same 2 cell lines, H1975 and HCC827, and we added the sequence mixture A and B in each of the cell line samples. And for the samples, we have 3 replicates. And we sequenced the same samples using 2 different technologies. One is the Illumina short read, and another one is the nanopore long read sequencing. And after generating the pure RNA samples, we mixed the reads from the pure samples to create the in silico mixture samples. And in our analysis, we performed the isoform detection using 6 different tools. And we aligned our pure sample and in silico mixture samples to the reference transcriptome and performed the quantification based on the reference. And then we performed the DTE analysis using 5 different tools, and also we compared 5 DTU tools. Then let's go to see the results. So firstly, I'm showing here the number of raw reads we get from the sequencing experiment, and also the number of reads that are actually mapped and quantified. You can see that we got actually quite a high number of reads because we used 5 flow cells for doing the nanopore sequencing. And here you may see that there is an Illumina sample that having a quite high number of reads. That is because in our first batch of the sequencing, we found out that this sample doesn't have enough reads. So we added another batch as a top up for that sample. So finally we got too many reads for this one sample. But if you look at the other samples, we even got more reads from the nanopore long read data than the Illumina short read data. So in this experiment, we get quite a high number of reads in both of the technologies. And as I explained that when using short read to perform transcript level analysis, there is some transcript level mapping uncertainty due to the complexity of the transcriptome and also the limited read length. So here we tried to quantify the transcriptome mapping uncertainty by using bootstraps when running salmon, and we use the HR function called catch salmon to estimate the over dispersion that are introduced during the transcript assignment. And as you can see on that plot, for both human transcripts and sequence transcripts, we found out that we actually get much lower transcript mapping uncertainty when we're using the nanopore long read data, the ONT data, comparing to the Illumina short read data. And here I showed an MDS plot. So for both our human and our sequence, we can see there is a clear structure on the on our different groups, and also they on the dimension 1. They are in the order of the proportion of the mixture. So this suggests that our data are showed the various structure clearly. And then let's look at the isoform detection analysis results. So firstly, we looked at the structure category of the isoforms detected by each method. And here we compare the isoform we detected to the reference. Sorry, excuse me. So here we consider the full splice match where the exon junction chain are the same with the reference, and we consider it as the known, while the incomplete splice match are having partially matched the splicing junction chain, but it's missing some of the junctions. Sorry. Well, for the novel transcript, there are novel in catalog and novel not in catalog and some other categories. So we expect the methods to have a high number of known transcripts, but also having a sorry, having a suitable number of novels detected. Sorry. Well, in our results, we found out that bambu seems to be working pretty well because it's having a high number of sorry. Because bambu is having a high number of full splice match and also having seems to be a suitable number of novels detected. But in this analysis, we found out that cupcake and flare are having a pretty high proportion of and the number of novel isoforms detected. And we think that it may be problematic because the proportion is too high and maybe they are false detections. Oh, sorry, false positives. And then we use the sequence where all the ground truths are known. I'm sorry. And we compare the transcripts we detected to the known sequence. And so by having the ground truths, we can calculate the precision and the recall. And in this analysis, we found out that bamboo is having the highest precision and recall. Well, in this analysis, flames and string type 2 are also performing well because they have a relatively high precision and recall. But we found out in this analysis, cupcake and talent showed quite low precision. And we used the sequence to do another analysis that we removed 25% of the known isoforms from the annotation to pretend that they are they are unknown. And in this case, our method should be able to remove the novel transcripts. And in this analysis, we also try to downsample our total risk to be 10% to see how the methods can perform when we having a low
Xueyi,
sequence.
would you like to just take a break for 10, 20 seconds just to have a water and then come? It's okay to pause for 30 seconds.
I just mute myself for several seconds. Sorry.
Yeah, so it's okay too.
Okay, I think I'm feeling better now. So let's continue. So in this analysis, we calculated the precision and the recall for the sequence that are kept in the dataset and removed from the dataset. And here we also found out that bamboo and string type 2 are outperforming the other tools by having a higher precision for both the kept and the removed transcripts in the sequence. And also string type, sorry. In this analysis, we found out that cupcake seems to have a low precision and recall. So in summary, for the isoform detection tools, we found out that bamboo and string type 2 are outperforming the other tools. And then we'll look at the performance of the differential transcript expression analysis tools. So firstly, we look at the overlapping of the DTEs detected by different tools on the long read and the short read data. And it's pretty good that the highest category comes from the DTE that are shared by all the methods. Sorry, let me just. So most of the DTEs are shared by all the methods on both the long read and short read dataset. But we also found out that the method Xueyi are detecting a high number of transcripts that are only detected by this one method. And we also find a high number of DTE transcripts that are only identified on the long read data. And then I used the in silico mixture series to estimate the power and the accuracy of the methods. So firstly, we look at the number of DE transcripts in different comparisons. So in this analysis, we found out that limma, HR, and DE-seq2 are performing quite similarly, while the method Xueyi are having a quite high power on the human data, while the method EBSeq are having a slightly lower power on both of the human and the sequence dataset. And then we looked at the inconsistency findings in the mixture samples. Because theoretically there shouldn't be any inconsistency, but here we found out that the noise-seq and EBSeq seems to have some inconsistency in the in-between mixture comparisons, but their inconsistent rate are all lower than the 0.05 threshold of the FDR. So they're all acceptable. And then we looked at the false discovery rate and true positive rate using the sequence because we know all the ground truth. So in here, we found out that the method noise-seq is not controlling the false discovery rate well in the comparison of the pure samples. Well, when we look at the true positive rate, we found out that limma, HR, and DSeq2 are all performing similarly, but noise-seq and EBSeq are having a bit lower true positive rate at the comparison of the in-between mixture samples. So overall, we think that DSeq2, HR, and limma are performing similarly well in these comparisons, and while in these three methods, HR is slightly controlling the false discovery rate better. And then let's go to the DTU analysis methods. So here we looked at 5 different methods. So firstly, I looked at the DTU transcripts identified in long read and short read data, and I found out that the overlapping is quite small, which means that when you are using long read or short read data to do your DTU experiment, you may having very different results. And then just skipping to this part that I used the sequence to look at the accuracy and the sensitivity. So firstly, look at the false discovery rate. We found out that only the method HR is controlling the false discovery rate well, while all the other methods at the comparison of the pure samples, they're having a false discovery rate higher than 0.1. But if we look at the true positive rate, we found out that HR is having a bit lower true positive rate than the other tools, especially for the last two comparison when the differences are relatively small. So in summary, for the DTU tools, we found out that there is no tool we can say that it's outperforming the other tools. And there is still a lot of space, and there's still a capacity to develop better DTU tools. So overall, in summary, we designed and generated a benchmarking dataset with plenty of good quality long and short reads. And for transcript level quantification, we found out that long read data is more suitable for this task with less mapping uncertainty and better accuracy. While for isoform detection, we compared 6 different tools, and we found out that bambu and string tie 2 are the tools that are outperforming the other tools, while string tie 2 showed better accuracy, but bambu showed better power to detect novel isoforms. Well, for DTE analysis, we found out that the DE-seq2, HR, and limma-voom showed similar good sensitivity, while HR outperformed in the false discovery control. But for DTU analysis, there is still a capacity to develop better performing tools for that task. And I found out that in our work, there are still limitations. So firstly, in our analysis, this work, we only used the Illumina short read and the Nanopore long read sequencing, while PacBio long read sequencing is also very popular, but we are not including it in this study. And because in our experimental design, we just choose 2 cell lines where we doesn't include any biological replicates. So the number of DEs detectable in our data are pretty high, which may not always be the case in real experiments. And also we are not including all the available tools for the benchmarking. And the performance of the tools are only measured with the specific version of the tools. But as I know, a lot of the tools have been updated after we did this experiment, and the performance may be changed. And also, as I heard that the nanopore sequencing accuracy has been improved quite a lot after we conduct this experiment. So currently the nanopore sequencing accuracy can reach 99%, I think, which is much better than what we have when we conduct this experiment. And finally, I would like to acknowledge Matt Ritchie and Charity Law, May, and Quentin, and all the other members from Ritchie Lab. And also, I would like to thank Gordon Smith and Pedro for providing help in this work, especially for using the catch salmon function from HR. I would also like to thank Jafar and Rory from WEHI Genomics Lab for generating the data, and thanks to Andy Winshen Chen for supporting me for doing the work last year while I'm working in his lab. And our paper has been published on Nature Methods last year, and there are a lot more details, and due to the time limit, I wasn't able to present all of the findings in this paper. So if you are interested to know more, please go and read the paper. Thank you.
Okay, everyone.
Thank you, Xueyi, for the great talk.